I'm very pleased now to introduce my esteemed colleague and collaborator, Dr. Jeannette Bennicelli, who is a scientist who's worked on diverse problems over her career. She is a star molecular biologist, cell biologist, and has carried out many of the key studies for this gene therapy clinical trial. And she's the newly identified director of the iPS cell core of the Center for Advanced Retinal and Ophthalmic Therapeutics. Jeannette Bennicelli.
Thank you very much, Jean, for that introduction. And today I'd like to talk to you a bit about what it was like, the challenges and the lessons that we learned moving forward from basic research to clinical trial development in our lab. I'd like to touch briefly on two issues. First, the design of our vector, and a vector which is very potent in delivery of the RP65 transgene. And then I'd like to talk about some of the issues that were new to us as basic scientists moving forward in clinical trial development. So here we see a schematic of our clinical trial vector. The expression cassette is shown between the AAV ITRs, which are shown in yellow. We have a hybrid CMB-CBA promoter, which allows for robust and ubiquitous gene expression. The promoter comprises an intron that has a synthetic branch point and a splice acceptor, and these also improve the expression of the gene. We also used a Kozak consensus sequence at the 5' end of the RP65 transgene, and this allowed for more efficient initiation of translation and also improved gene expression. And finally, in the backbone of the vector, we used a lambda stuffer, and this increases the size of the backbone to greater than 4.7 kb, and this prevents reverse packaging from the AAV ITRs. This slide shows the importance of formulation in the accuracy of AAV dosing. AAV adheres to inert surfaces, including the storage vessels, the filters, and the vector administration devices. And we found that the use of surfactant in our formulation allows for a more consistent and predictable delivery of a given gene dose throughout the trial. We used pluronic F68, and on the right you see a diagram with a vector administration device, and in panel A, in the absence of surfactant, you see that a large amount of material is retained inside the device, whereas in the presence of surfactant, we have a more accurate delivery. These are some of the data that support the use of pluronic. Whoops, I didn't mean to do that. That support the use of pluronic in our formulation. On the top panel, we're looking at qPCR and recovery of AAV RP65 after passage through 3 different administration devices. On the y-axis is the titer, so the percentage of a vector that we're recovering. And the first 2 data points show the recovery of vector pre-device, both in the presence and absence of surfactant, and you see that we have 100% recovery as expected. The next 3 data points show the recovery after passage through the 3 devices in the presence of surfactant, and you see that we have virtually all of the activity recovered, whereas the last 3 data points show what happens in the absence of surfactant, where a large amount of material is lost to the device. The second panel shows the recovery of functional activity after the virus is held in different administration devices. And here we're looking at AAV-LAC-Z, and we're looking at functional activity in vitro and transduction of cells. So the first column shows the activity of the virus pre-device, and then the next 2 columns show what happens when the virus is held in administration device A or B for a period of 2 hours in the presence of the surfactant, and you can see that we recover virtually all of the activity. When the material is held in the device for as long as 20 hours, you can see that there's really a negligible loss of activity in the presence of surfactant. So these data show the importance of having surfactant in our formulation. So in this last slide, I'd like to touch briefly on some of the issues that were new for us as basic scientists moving forward into clinical development. These are regulatory issues, timeline issues, and clinical assay development issues. In terms of the regulatory issues, Jean touched about this when she was talking at the beginning, are the requirements for reporting of the documentation of the experiments, which were not totally unexpected, but we did not really realize or appreciate that these same reporting requirements would be required for any ancillary studies that we wanted to do. And so I think if we had known this from the beginning, we would have planned things a little differently, but we had to learn about filling out all those documents that Jean was talking about. In terms of timelines, we needed to balance the need to meet regulatory deadlines with the need to optimize our materials. And this was particularly important at the beginning parts of our development when we were working on optimization of our vectors. And so we were trying to meet our regulatory deadlines, but also trying to make the best vector possible. And this required a lot of flexibility in these regulatory deadlines so that we could meet them and also have the best product available. We also learned that we needed to plan ahead for some of the assays that we were going to be needing during the clinical trial and later phases. And in particular, we needed assays for potential immune responses to both the transgene and the vector. We needed a potency assay, and we also needed a biodistribution assay. And we had to develop these assays way in advance with SOPs, with assay validation. And also, very importantly, we needed to find a source of standardized materials that would last for the duration of the assay, which in this case is 15 years. So that was not a trivial matter. So in summary, these are some of the issues and some of the lessons that we learned from the basic research lab.
Thank you, Jeannette. Next, we have Dr. Dan Chung, who is our Director of Outcome Measures on clinical trial development. And Dan is trained as a pediatric ophthalmologist, and we're lucky to have him here today.